Hi. So, what do you think about when you think about acid-base reactions? You could take a moment and try to remember what you've learned about them and sort of what you associate with those terms, because we're going to be adding to your pre-existing understanding of acid-base reactions. So do acknowledge that you've probably already learned something about them. And we're going to be building on those same sets of ideas. So one of the things that students often think of is pH, where those high pHs are basic and the low pHs are acidic. We almost never use pH in organic chemistry. Instead, when we talk about acids and bases in OChem, we're usually talking about Bronsted-Lowry acid, or BL acids and bases, where the acid is donating a proton and the base accepts a proton. We use some specific terminology that might be a little bit different than what you've seen before. We'll be talking about the protonation state of a molecule, and that's if an acid is in the acidic form or in the conjugate base form, or if the base is in the basic form or the conjugate acid form. And that's something that's really important for biomolecules, which we'll look at in a second. Bronsted-Lowry acid-base reactions also play a role in almost all organic chemical reactions, and we'll look at that a little bit at the end of this introduction. So protonation state and enzyme activity. We'll look a little bit more at this example in class, but what we're looking at here, just to kind of start you off, is these lines here. This line is meant to be the active site of an enzyme called acetylcholinesterase. And then if you could imagine, the outside of that is the rest of the enzyme that provides important structure. And the inside area is where acetylcholine goes into to be hydrolyzed after muscle contraction. And this is an essential activity that's occurring in your body all of the time when you move and when you breathe. And so what happens is acetylcholinesterase goes into the active site, and by hydrogen bonding and ionic bonding, it kind of finds its correct position, which is something that we'll look at in class. And then it reacts with the serine over here to be hydrolyzed. And so, whoops. So I'll remove some of these boxes here. The protonation state, in other words, that this is actually a deprotonated carboxylic acid. We call it a carboxylate. It's important that it's negatively charged. If it was neutral, if it had a proton on it, then it wouldn't be able to form an ionic bond with the acetylcholine when it comes into the active site, and it wouldn't stay in there. Similarly, if the tyrosine had its proton, or if it was deprotonated and it was negatively charged, again, acetylcholine wouldn't stay in the active site. And the OH, again, the protonation state of the OH is key. It can't be deprotonated, in other words, be a negatively charged oxygen, or it couldn't be protonated, which would give this a positive charge. If either of those were the case, then the reaction that needs to happen wouldn't happen. And so these protonation states are really important. The protonation states, and here's one of the times that we'll actually talk about pH, are determined by the pH of the solution or the environment. And so enzyme activity is really determined, in other words, like how fast the enzymes work, is determined by the pH of the environment. And so enzymes will have optimal pHs, times that they work, or pHs under which they work optimally, and that's when the protonation state is all correct. Lots of our optimal pHs are around 7, because that's And that's where our body, the physiological pH is. So one of the things that you want to be able to do when we look at chemical reactions or its structures is you want to be able to identify acids and bases. And this really is the same thing that you've done in general chemistry or other chemistries or other chemistry classes that you might have taken. And what we're looking for the Bronsted-Lowry acids and bases is we're looking for a proton. And I say proton because the thing that's going to be transferred is just a proton. The hydrogen leaves its electron behind, and so it's only a proton that's getting transferred from one molecule to another. So if I look at the starting materials, the reactants, and the products, I can see that this proton that's in red kind of jumps from the water over to this is terbutoxide, and it protonates it, and we wind up with the alcohol and hydroxide as our products. And so this is the acidic proton, and we try to be really specific in our organic molecules and not just call the whole thing an acid or the whole molecule a base, but be really specific about the proton that's acidic, or because it's being transferred over to this negatively charged oxygen, we would say that this oxygen is basic. So we have our acid, our acidic proton. This oxygen is the base. And you can see that it's transferred, that proton's transferred. And then we are left with the conjugate acid. So now this is tert-butanol, and the base then becomes the conjugate acid in the product, and the acid becomes the conjugate base in the product. And indeed, the reaction could go in the other direction, and it is at equilibrium. And so we would expect that to occur. And so the conjugate acid would act as the acid in this proton here. The conjugate base would be this oxygen. So one of the this is really one of the first times that you'll be showing how a reaction occurs. And we call that showing a mechanism. A mechanism is the order of bond breaking and forming over the course of a chemical reaction. They're hypotheses. We can't see them directly, but we can be pretty sure through lots of different experiments how things happen, what order bond breaking and forming occurs in. And we indicate them with this curved arrow notation. So here's one curved arrow, here's another. And we call we could also call that electron pushing arrows. And you'll see me use both terms. I think Klein uses the curved arrow notation. Just as a reminder, looking at this reaction, make sure that you can identify the acid, the base, the conjugate acid, and the conjugate base. So hopefully that's what you came up with. And you do want to be able to start to write these curved arrows to show our mechanisms. You could also use molecular modeling, where you're actually seeing the molecules changing as a way to indicate how a mechanism occurs. But when we're just writing on paper, we use this curved arrow notation. So note that the curved arrow starts at the electron-rich site. So that means a lone pair. Let's see here. Unroll. Yeah. The high electron density place, a lone pair, or negatively charged species. We'll talk about nucleophiles soon, but just an electrophiles soon. You can ignore those terms for now, but maybe tuck them away. We'll come back to them in a little bit. We have this lone pair, it's electron rich, and that is where the arrow should start. And you can't have your arrow off above it or below it or just kind of in the general area. It really has to start at that electron rich site. And This sometimes is a little bit confusing in acid-base reactions because sometimes students just want to show how the proton moves. They're like, oh, the proton jumped from the acid to the base, so that's how I should show my arrow. And that's a really common mistake in the beginning. That's actually not how we use electron pushing arrows. Electron pushing arrows show the flow of electrons, not the flow of protons. So we have our electrons here, and they are forming a bond. That's what this red arrow means. They're forming a bond with this proton. And at the same time, the electrons in the oxygen-hydrogen bond stay with the oxygen. And so that gives oxygen 3 lone pairs and 1 bond. So in the product, it's negatively charged. This oxygen here, it has 2 bonds and 2 lone pairs, and so it's neutral in the product. When you are drawing acid-base reaction arrows, you should have those 2 arrows, and the first one should be from the lone pair to the proton, and the second one to show how the bond becomes a lone pair. So here's a second example of this, and that is where we have, in this case, our organic molecule is the base, and our acid is hydronium. That's water with an extra proton stuck on it. And what happens in the first step is an acid-base reaction. We also see it referred to as a proton transfer. You can see that the arrow starts at the lone pair, shows a new bond forming to the proton. At the same time, the bond between the oxygen and the hydrogen becomes a lone pair on the oxygen. As we see here, we get our product. Now, this step is actually not an acid-base reaction. It's an elimination of water or dehydration. And overall, this is a mechanism for an elimination reaction, a reaction that you'll learn about later on in the class. It's shown here just to demonstrate how acid-base reactions are a part of many reaction mechanisms that we're going to see throughout this class. So we have this elimination step, and you'll just have to trust me for now. That gives you your positively charged, this is a carbocation. It's a carbon with 3 bonds, a positive charge. And then in the second step, we have the lone pair of oxygen acting as our base. It deprotonates the proton, and we wind up with hydronium again. And the 2 electrons that are in the carbon-hydrogen bond then actually in this case become a pi bond, as we can see here in the product. And so this is a catalyzed reaction, meaning that we start with hydronium and we wind up with hydronium. And the thing to focus on here is how the arrows are being used in the acid-base parts of the mechanism.